Emily Stevens and Max MEHLMAN are both PhD candidates in psychological and brain sciences program at Dartmouth College, and their research is absolutely fascinating. Max works on spatial, directional processes in the body that help tell us where we are in the world, upside down, or back to front, left and right. And people who say they have a good sense of direction might be referring to processes in their brains that are particularly adept at helping them decide which way they're going. Emily's research is more focused on the chemistry in the brain and how that responds, particularly now, to the effects of certain antidepressant drugs and what they do to those processes in the brain. That is a super basic layman's assessment introduction to their research, but they're both very accomplished and have won several awards, have published, and are very used to talking about their research to public audiences. So without further ado, thanks very much for agreeing to present and chat about your research tonight, and thank you again for coming.
Okay, well, thank you, Amanda, and for the co-op. This is a fantastic place, and it's great to be here, and we're excited so many people showed up. So today we're going to tell you, I think the title is How the Brain Works. We're probably just going to give you some insights into how the brain works, because that's kind of an ambitious thing to tackle that admittedly we don't really fully understand. So I think we're going to start by just kind of giving you some overview, kind of background information about some basic properties of the brain. And then afterwards, we're going to kind of each go into our own specific research and tell you a bit more specifically about the types of work we do, like Amanda alluded to in the beginning. As much as we can make this a discussion, the better, right? So feel free to just shout out or raise your hand if you have any questions or if things are unclear. And I'll be happy to clarify things as best as I can. Okay, so the brain, obviously this big hunk of tissue inside of our skulls. The easiest way, in my opinion, to think about the brain is a computer analogy works really well. The brain processes information much like a computer does. So I like to think of the brain as kind of working in these three steps. It perceives the world, it kind of has this sensey thing that it does. Then it kind of processes that information, it tries to make sense of that, so it has this thinky thing that it does, and then it needs to respond, right? It needs to move our muscles to kind of respond to what's going on in the world appropriately. So it does this doey thing. So you have sensey, detect the world, thinky, that's like cognition, that's what the mind is doing, and then doey, we have to control our body. So you can kind of think of the brain as being, here's a really poorly drawn brain, and then there's kind of input into the brain, some processes are occurring in there, and then there's some output. So the input is coming from our environment, and we detect that with our sensory organs, so our eyes, our ears, our nose. So the typical 5 senses you think about, that you learn about in grade school. There's a lot of other senses that we don't typically think about as much as vision or audition. For example, our vestibular system provides us with this really good sense of balance. You know if you're standing upright or if you're laying on your back, right? So we have this kind of sense of our body position. We have sense of the passage of time, right? We know if something happened an hour ago or 3 days ago. So there's a lot that we're detecting in the world. That's all from our sensory organs. And then the brain is going to kind of make sense of this stuff. I'm seeing like some tan over here and it has these different surfaces. Oh, it's a chair, right? So that's what the brain is going to be doing during that kind of thinky component. And then finally, oh, maybe I'll go and sit down in the chair. I need to like lower my legs, bend my knees. That's going to be the doey component, right? So that's kind of a very easy way to think about brain function. A really important principle about the brain is something referred to as localization, and that means that different functions are localized to different parts of the brain. So we can see that in terms of the sensory systems. Our eyes are sending that input to a very specific part of the brain that's involved in processing visual information. Our ears are sending information to another part of the brain that's dedicated to process auditory information. Our vestibular system projects yet to another part of the brain. So the brain is kind of all these different modules, these different components that are all interconnected, and they're all kind of processing different types of information. So we have different parts of the brain that are getting all that sensory information, and then we have other parts of the brain which are referred to as like association areas, where all those different singular senses, all that input will kind of converge into parts of the brain that then can make more like holistic representation. So if a dog is barking, like the visual stimulus of the dog and the bark and my memories of what dogs are, all that stuff kind of comes together in these association parts of the brain, which tend to be these cortical regions, generally the frontal part of the cortex. So those are kind of like the two kind of background points I wanted to make. There's like this input, processing, output in terms of the way the brain functions, and then in the terms of the way the brain is structured, we have this principle of localization, where different parts of the brain are dedicated to different types of functions. And the initial insight into this, into localization, came from comes from situations in which people had like focal brain damage, and they presented very specific types of like different consequences. So there's a really famous example of this guy, Phineas Gage, who's actually, he worked at the Hanover Inn, so it's kind of this cool regional tie to this story. But Phineas Gage was working on railroads, and he had a tamping iron, and he was tamping charge down into a hole, and it ignited, and it shot this large iron rod back at him, and it actually somehow went through his eye and shot through his head and damaged the frontal part of his brain, the frontal lobe, and he miraculously survived. But afterwards, his personality had wildly changed. He was like a totally different person, and he had no self-control. He used to be a very like polite guy, but after this, he was very rude and kind of aggressive. And so this led clinicians at the time to think, oh, maybe our self-control, maybe our personality, maybe that kind of resides in this front part of the brain that happened to get damaged in this individual. Similarly, there's another famous patient who went by the abbreviation HM for a while. Now we know his identity, this man named Henry Molaison. And in the late 50s, he had part of his brain removed in an attempt to cure a really severe form of epilepsy. And so they removed this part of his brain called the hippocampus, and then afterwards his epilepsy was cured, but the clinicians quickly realized that this individual could no longer form memories after this procedure. And he kind of was frozen at the moment that his surgery happened. So he kind of remembered his childhood, he remembered everything that happened, but he couldn't form new memories, like moving forward in life. So you meet this guy, you talk with him a little bit, if you left the room then came back in, he wouldn't recognize you. So this is another example where scientists and clinicians realize that when this very particular part of the brain is damaged, there's this very particular consequence. So again, this is another example of finding localization in the brain, specific functions kind of residing in specific parts of the brain. And a lot of modern neuroscience is kind of following up this kind of initial preliminary insights into localization, and now we have much more powerful tools to bring to bear on these questions, and we're really trying to map out functions in the brain to really say, this function is in this part of the brain, this other function is this other part of the brain, and we want to know how those different parts are connected and how they interact. So now Emily will describe, kind of zooming in to a bit more kind of microscopic detail, what's going on at kind of the cellular level in the brain.
So like all of your other organs in your body, the brain is made up of cells. And so cells are individual units, like biological components, that kind of subsist on their own but can work together to create a larger organ. And so in the brain, there are multiple types of cells. There are neurons, which people may have heard of before. We usually call these brain cells, kind of like the same thing. These are the types of cells that perform a lot of the information processing, kind of this thinky part that Max was talking about. The neurons are the unit in the brain that do a lot of that processing. There's another type of cell in the brain, this large subgroup of cell called glial cells, and actually I know almost nothing about them, but they are, some people have called them the support cells for the brain, but there's a lot of research that shows that that's actually not super accurate anymore. We know a lot more about them and seem to have discovered that they do many, many things and may even be a part of this processing of information. And so that's kind of a whole other can of worms that we could talk about another time. But the glial cells are responsible for doing things like they're part of the immune system that takes care of the brain. They regulate some of the ion concentrations in the brain. They help signals in the brain travel faster from neuron to neuron. So these are the kinds of the supportive and maintenance things that these types of cells could do for neurons. But I focus on what neurons are doing, because as I said, neurons are the unit that's doing the majority of that information processing. And so a neuron, a very classic graphical representation of a neuron, I'll show you kind of here, this kind of cartoony version, starts with the neuron's cell body. And the cell body is the main area of the cell that shares a lot of the common structures that you would find in other cells throughout your body. And once I finish drawing it, I'll tell you what those are. Neurons are very much like all other cells in your body. They have a central core region called the nucleus. Right here, the nucleus is the part of the cell that contains all of the genetic information. So all cells in your body contain the same genetic information, and that is stored in a part of the cell called the nucleus. Neurons also have these little energy factories called mitochondria. These, as I said, make energy for the cell. They also break down oxygen. They perform kind of the respiration or the breathing that a cell needs to do, taking in certain nutrients. There's a few other organelles like little ribosomes. These are small little protein factories. Ribosomes make more proteins for the cell, and proteins are kind of the materials that cells use in order to function. So proteins inside of a cell are different from the proteins that we ingest, but at the molecular level, they're quite similar. They're things that allow the cell to kind of function and do what it needs to do. So ribosomes make those. There are many other organelles that are a little bit, probably don't need to go into explaining here, but as I've just explained, all of these things are common to all types of cells. But if maybe you think back to a biology course you've taken in the past or other type of science course, one thing you can start to maybe remember is that most cells actually look kind of just like this. Just a kind of amorphous blob. That's different from what we typically think of when we think of a neuron. So a neuron is what's called a polarized cell. So it has one part of the cell that's doing a specific function and another part of the cell that's doing another function, and they're separated spatially. And the polarization that neurons have to them is an important part of what allows a neuron to do kind of very, very low-level information processing. So before I get into what that actually means, I should explain that neurons themselves are electrical. They're kind of like little batteries, so they can send little electrical impulses to each other all the time. So at any moment, any function that you're doing, even during sleep, controlling your breathing rate, digestion, all of these things, those are mediated by these electrical impulses that neurons send to each other, which are called action potentials. I might just call them spikes in the future, and that's because when we record these electrical impulses on a computer, on a recording setup, they look like little spikes, these little positive and then negative advancements of a line, kind of like this, kind of like that. That would be an action potential we would record on a computer. And so these electrical impulses begin in an area of the neuron that's very close to the cell body. This is called the axon initial segment. And this is a very important part of the neuron. This is where electrical activity comes together and kind of grows to a certain threshold, and then gets sent down the axon, which is this part here, so that it can be transferred to another cell. And so you may be asking, what are these things coming off of the cell body? They look a little bit different from the axon. These are these projections off of the cell body are called dendrites. And they're called that because they kind of look like the branches of a tree. And dendrites root comes from the Latin, correct? Word for tree, Latin or Greek, probably Latin. And so, so these electrical impulses that are sent from cell to cell get received largely in the dendrites. So one cell will have an electrical impulse that it starts here at the axon initial segment, sends down its axon, and then gets transferred to another cell's dendrites. And then in that second cell, that electrical impulse can come together and either send off to another cell or stop. Kind of that's where the information processing layer comes in, and maybe we won't go too much into that. But one really, really important part about this form of communication, this electrical communication, that's kind of like electricity moving through wires in your house to power anything electrical like lamps and televisions and stuff, is that this actually isn't direct. It isn't a direct transfer of electricity from one cell to the other. You may have heard of something called neurotransmitters. These are chemicals in the brain that actually continue that electrical signal from one cell to the other. So neurons in the brain don't actually touch each other. They're pretty close, but what will happen is that the electrical impulse will travel down an axon and will cause release of a neurotransmitter into the little space between cells called a synapse. And then the chemical will then move from one cell to the other and induce that electrical signal again. And so collectively, we refer to this process as electrochemical communication. So you start with the electrical signal in the axon initial segment, moving down the axon to the synapse, where neurotransmitters are released onto dendrites to then continue on in the signal. There's a lot of layers, but in the very basic form, you have electricity that gets translated into chemicals, which translates back into electricity. Kind of this up and down system. So there's many different types of neurotransmitters that can be used by different cells in the brain. And I'll get into one neurotransmitter that I study. It's called serotonin. You may have heard of it, but there's actually a lot of different neurotransmitters that may or may not have heard about in your daily life. So there's a neurotransmitter called dopamine, which is highly involved in processes underlying certain mental health disorders, including addiction, and also neurological disorders like Parkinson's disease. So in Parkinson's disease, dopamine is made by a very small set of neurons, actually a surprisingly small number of neurons, in a specific area of the brain called the substantia nigra. It's called this because when you stain brain tissue, or actually when you just look in the brain without even staining it, this area of the brain is black, and so substantia nigra means black substance. So it's kind of just a cool thing. You can just see that right in your brain. And so this is the area where I said dopamine is made. And if you have selective damage to the neurons in that area of the brain, you have reduced dopamine throughout the rest of your brain, because that's where it's being made. Getting rid of those cells is going to reduce the amount that you have everywhere else. And so reduced dopamine in your brain can lead to a lot of these the symptoms that are associated with Parkinson's disease, like resting tremors and difficulty with initiating movement. There's other neurotransmitters that are involved with some neurological diseases, like Alzheimer's disease has been associated with acetylcholine, which is another neurotransmitter. Acetylcholine reduction in the brain has been thought to be related to Alzheimer's disease, but it's actually still something we don't understand very well and are still doing a lot of research on, and kind of trying to figure out why in some patients reduced acetylcholine is related to their disease progression. There's also a neurotransmitter called norepinephrine. It's very similar to adrenaline, which is something that you probably have felt through your body, kind of an adrenaline rush. Norepinephrine is the neurotransmitter that's involved with changing your body and changing the kind of state of your body in response to external stressors. So when norepinephrine is released, what happens is your body prepares for, this is kind of the fight or flight response. It prepares to flee or fly away or run away. So if you are walking on a trail and you encounter a bear, you probably feel that kind of pit of your stomach, like, oh no, terrible feeling. That's norepinephrine helping you kind of prepare your body to flee. And so, as Max said, a lot of these neurotransmitters are going to be localized within the brain. This is going to be similar to the localization of function. And as I alluded to with Parkinson's disease, you can have selective reductions in some of these neurotransmitters that lead to specific disorders. And so the way that we study a lot of these different systems is by using multiple different types of recording techniques, because if this is an electrical system, we can record the electrical activity, kind of like if you were trying to understand if you had a fuse that was out in your house. You're trying to figure out which fuse you needed to replace. You may be turning on or turning off different lights and checking on different fuses in the fuse box and trying to change things. We do a similar-ish thing where we investigate different parts of the electrical system and see which parts work at certain times during certain behaviors. If we turn on a light in the living room and this fuse is turned or has been blown, then the light actually won't turn on. Okay, we can do something kind of similar if we're looking at an animal that has a lesion, or which means kind of a selective damage in a certain part of the area. What type of behavior is gone? That's something that we can. That's part of how we can learn more about the brain. We can also learn about the brain by recording individual neurons. So we can take brains and look at individual cells within the brain and record their electrical activity and put different neurotransmitters next to them and in the solution in which they're being kept, and we can say, oh, what does this do to the electrical activity of this cell? And so that's kind of getting into the area of research that Max and I do. Before we move on, does anyone have any questions about what we told you? There's a lot that we just threw at you. Yes, please ask questions.
Sure. You talked about the communication from one cell to another via the axon, the neurotransmitter, and the dendrite. Do any cells have any self-communication? Do they communicate? Any of them communicate with themselves?
Yes, definitely. So the many layers of complexity that exist in the brain come from the ability of neurons to fine-tune their connections with each other, the types of neurotransmitters that are involved with those connections, how many connections are made, and what types of cells communicate with each other. So there are different types of cells in the brain. Some release a neurotransmitter that always causes more action potentials and more electrical activity and sends signals along more. And then there are some neurons in the brain that release a neurotransmitter that does the opposite and reduces the amount of neurotransmitter, or reduces the amount of electrical activity in the next cell. And so in a situation, if a cell wanted to send a signal back to itself, it could either do that by releasing neurotransmitter onto itself, or to a neighboring cell that's actually maybe a type of cell that says, yes, do more activity, or a type of cell that says, no, don't do so much activity. So they can regulate themselves either immediately onto themselves or by talking to their neighbors who can help them out with that too. So the complexity of synapses is something that is very, very widely studied. So synapses, which we talked about, are the connection between two different cells that are formed by the axon talking to the dendrites. Those are dynamic. They can change with learning. They can change as your memory develops and changes. They are something that develop and change throughout your entire life. There are some that are much more hardy and will stay around a lot longer but may eventually go away. We're actually really unsure about how the stability of synapses leads to long-term memories and these types of things. So a really cool example of this is, so there are squirrels that live in the Arctic Circle, Arctic ground squirrels instead. So they can go into a state of hibernation or torpor where they have very, very low metabolic rate, which means that they're not using a lot of energy, they're not eating. Right? It's very similar to bear hibernation, right? But they are doing this in more extreme temperatures than what might be found around here. Very, very, very low cold, sub-zero Fahrenheit temperatures. And in this situation, it's actually totally not understood how these animals can survive, because as a mammal, you need heat to keep your tissues healthy. somehow they survive, and they have this very cool thing that happens in their brain where the dendrites and the synapses and the axon all shrink back into the central cell body, the soma, which is another word for cell body. They shrink back in and get really short, and they kind of just simplify. The neurons become more like this. And at that point, that kind of happens throughout the duration of the hibernation, and then when the animal wakes back up, they become more complicated again. They stretch back out, they form new synapses, and it's not like the animals totally forget their life before. All their memories are there, they're able to walk around and forage for food. I'm not 100% sure, but I think they're able to remember things that they did before, like, oh, I stashed my nuts over here, I will go find them. They have these memories, and it's totally unclear to science how that works, because we really do think of synapses and the specific arrangement of dendrites and axons as being very important to the way that memories work and learning works and kind of how we function. So it's kind of an interesting thing. Yes, please.
Can a single cell body produce more than one neurotransmitter, and can it respond to more than one neurotransmitter?
Oh, these are excellent questions. So, to your first question, can a single cell produce more than one neurotransmitter? Very recent evidence shows that yes. There are certain cells that actually in the same, so neurotransmitters are released in these little bubbles that come off of the end of the axon, very similar to if you were to blow a bubble out of a little bubble stick. They kind of just come out of there and then release neurotransmitters. In those same bubbles, we think that more than one neurotransmitter may exist, which totally goes against everything that neuroscientists have thought for a very long time. And so that's pretty exciting and pretty new. We're really not sure why that is, if that's something that is specific to certain types of neurotransmitters. Maybe only one type of neurotransmitter can pair up with other ones. We're still finding all the time many examples of this where we previously had thought that was totally not possible. And to your second question, can cells respond to more than one neurotransmitter? Absolutely. This is another layer of complexity. So if a neurotransmitter comes in, say dopamine here, dopamine as a chemical, in order to say anything to the cell, needs to hook up with a specific type of protein called a receptor. And so this receptor hangs out on the surface of the cell, and dopamine comes in and hooks up to it, forms kind of a bridge or kind of chemically connects to it. And then because of that connection, the cell learns, oh, dopamine's around, and does whatever it needs to do in response to dopamine, which is always different depending on the cell and the receptor. But this cell could also have a receptor for serotonin, and they might even be really close to each other. They might be right next door, they may be right across the dendritic aisle, or they could be in very different parts of the neuron. And these two things may do opposite things to the cell. Maybe dopamine, talking to this cell, says, yes, send more action potentials, send more information, do lots more. And maybe this one, serotonin, comes in and says, no, don't do that. So then maybe, in this situation, if the whole system that this cell is a part of wants to send more information, dopamine is released so that more information is sent. But then maybe it gets to be too much and it says we need to slow down. So then serotonin comes in and it's released and says, okay, let's slow down. So there's this different levels of regulation. To make things more complicated, depending on the receptor, it can have different effects too. So that's actually kind of very specific to the type of work that I do, and I'll explain that in a little bit more detail later. Other questions?
Just out of curiosity, that's a healthy looking cell, right?
Yeah, yeah, so this is basically a healthy cell.
If a person has dementia, will it look a lot different?
That's a great question. Do you know if individual neurons look different?
Well, with things like Alzheimer's, there's certain types of proteins accumulate in the cells and the space around the cells. But there's a lot of different ways that the brain can kind of go awry. So there's some cognitive and neurological disorders associated with damage to specific cells, so like the shape of the cell or the functioning of individual cells is disrupted, whereas other disorders may be specific just to one specific type of neurotransmitter is ineffective, or certain connections between regions are disrupted. So you can kind of disrupt the function of the brain at the level of individual molecules, at the level of individual cells, or at the level of like the interactions between cells.
Yeah.
But this is a pretty healthy looking
Yeah.
cell.
I will also say that I drew this as kind of the classical cartoon that you might see in a textbook. Actually, so the axon is like very large here. Most axons are very, very small and very long. So you have axons from some of your cells that extend all the way from this part of your brain right here down to your toes. These are called Betz cells, named after the person that discovered them, I think, Betz. And this axon is really important for sending information about moving your bottom limbs. And so this axon it can be multiple feet long, and especially very tall people. But some of them are also very short. They can send axons directly to their neighbor too. So, yes, please.
Does that travel down your spine?
Yes.
And
Yes.
out of your spinal column?
Yes, exactly.
So it's not necessarily a nerve, it's an axon.
Yes, actually, so the word nerve usually refers to many axons bundled together. So, if you were to have some sort of anatomy class and they showed you in a spinal cord all the nerves, those are actually many, many, many axons. Axons are generally microscopic in most animals, specifically in humans. But when you bundle them together and wrap them in kind of this kind of like an electrical wire wrapping, kind of like the insulating plastic or rubber, kind of like that, then that's what you would call a nerve. And those travel down your spinal cord. And then actually, the peripheral nervous system, which is defined as all of the nerves and neurons and axons that exist outside of your brain and spinal cord, so all throughout your arms and your legs and throughout your torso, all the nerves and neurons there are your peripheral nervous system, and then you have your central nervous system, which is your brain and spinal cord. But yes, it would travel throughout. There's two questions. I'm going to go right behind you.
You can go right ahead.
If what you're talking about now is the physiology of the brain, what happens when the mind comes up with concepts, ideas, and a desire to execute something, but it has to facilitate, it has to send messages to the physiology part of the brain to the action? How do the two connect, the thought and the action part?
Yeah. So that's a super tough question. So, yeah, no, but it's a great question. No, you shouldn't. It's a great question. So I think kind of the root of that question is, what is the difference between mind and brain? And I think that almost every neuroscientist would have something different to tell you about that. And I think that depending on the level of mind that you're thinking about, there are different answers. So your brain controls all of your what are called autonomic functions. So your breathing rate, your blood pressure, your heart rate, digestion, all of these things are controlled by your brain. And in a way, that is your mind, because it's a part of the structure that is your brain and that controls all of your cognition. But your brain also controls things like dreams and ambition and motivation and all of these very complex, abstract concepts. And that would be something that I don't think any of us could say, that is located here. So when we get rid of that, we don't have motivation anymore. It's much more diffuse and abstract. And so that level of mind, I think, is much more intangible. Do you have a better answer than what I just said, Max?
Well, yeah, it's like, in a way, it's very, it's almost more of at least at the current state of neuroscience, it's still, the question's still more in the realm of philosophy at this point, in terms of like the mind-brain duality, how like our thinking cells, our self-identity, all the human experience, this crazy thing we go through, how do we separate and how do we map that onto a physical organ that's a bunch of cells and proteins and different molecules that are physical things that are shuttling around. I think most neuroscientists would just say that cognition and the mind is an emergent property from the brain, and somehow all this electrical activity going on, the physiology of the brain, this stuff existing in the real world, that generates thoughts and emotions and identities and drives in our mind. And but if we're getting there, we're only beginning to scratch the surface of how consciousness can arise from the biology of the brain.
Okay, because one of the things that I've learned from some of those things that I've personally studied on my own is that when you have a level of intellectual intention, you can actually redirect the outcome of healing your own body versus going, let's the AMA route. You choose to heal through your thoughts, through your emotions.
Well, and that's a great.
There's a connection there,
right,
a very
and,
deep connection.
Right, and so it's bi-directional. It is, the body is, the body and the brain may generate the mind, but our own thoughts, right? We can think of, like, if you think about something very upsetting, your heart starts to race, you may get a little sweaty. Mm-hmm. That shows you how, like, our mental processes, our thoughts do flow the other way, right? Our thought, if we pick up the phone and get really good news, or pick up the phone and get really bad news, our body, our physical cells will react much differently to those same situations. So yeah, the brain is kind of generating thoughts, but thoughts also do have this influence back on our physiology. But there's still this kind of black box. It's still very unclear. How do you go from molecules and biology in the real world to cognition and thoughts and hopes and dreams, and then what is the actual, like, conduit that's allowing these two things to influence each other? And I would say that's probably the biggest mystery aside from, like, what is the universe and, like, where does cognition and consciousness come from? I think that's probably, like, that's why I'm drawn to this field, because I can't think of a bigger question to ask than, like, how do thoughts come out of action potentials?
That's why I went to grad school. Yeah. I went to my undergraduate advisor and I said, I would like to know where thoughts come from. And she goes, yeah, me too. She go to grad school. So then I did. so yeah, it's a really complex question, and there's a lot of ways to think about it. And I think Max did a really nice job at explaining kind of how we, as neuroscientists, kind of think about it.
And it's, well, and the way that we design experiments, you need to control so many things, and things need to be so rigidly defined. And a lot of the work, especially when we're interrogating the physiology of the brain, is being done in animal models, like rats. So in terms of just the experiments that can be done, we're kind of limited. We're still trying to figure out, like, how does a brain hear? How does a brain see? How does a brain remember that, oh, I got, like, some really good food over in that corner of this maze last week, I should go back there. We're kind of still at that level. So getting to consciousness is gonna take some more decades or centuries.
Yeah. I think we have a couple more questions.
So just related to this lady's question, I mean, in this world there are people who would have different abilities. I would not be able to study what you're studying. There's certainly, there's certainly some difference between you and I. So is there any sort of physical or physiological difference that neuroscientists have found between very intelligent people and not so intelligent people, or ones who kind of peter out at a certain
like, is there
level?
a maximal, right?
Yeah.
So, as a very extreme example, Albert Einstein's brain was evaluated after he passed away, and he was found, please correct me if I'm wrong, to have more synapses than an average brain. However, what that came from is a really important question. So was he born that way, and that allowed him to do a lot of the things that he did in his life and ask all of these questions? Or did those many number of synapses arise because he spent a really long time thinking about things and challenging his mind and reading a lot and having discussions about very abstract concepts? We don't know that, and we can't know that. there are genetic factors that may predispose someone to being able to read at a higher level, or have a certain ability in math or things like those. In general, though, I would say that those kinds of genetically specific abilities are in the minority. So I don't think it would be fair to say that there's a math gene, and if you have it, you can do math, and if not, then no. I think a lot more of what goes into individual differences with things like intellectual capacity and personality have a lot to do with experience and especially early life experiences. So learning and being in an environment early on that challenge you cognitively and bring you new experiences. Though I think that there are theories about how different physical parts of the brain, like your synapse number or complexity of other elements that make up the brain, may be related to intelligence. But I don't know.
Yeah, like you said, it's kind of touching on this nature versus nurture debate, and it's really difficult to say, is this person's brain, or does this person have this skill because they have this brain feature, or do they have that brain feature because they have the skill and they've been, like an example is like if you're a pianist or a violin player and you have to have very precise motor control of your fingertips, the part of your brain that controls your fingers will actually kind of expand and get more elaborate because you've spent so much of your life doing all this very fine motor stuff. Similarly, if you're a drummer, your ability to it would be called temporal processing, your ability to kind of time and like keep a rhythm gets like really, really good, and the parts of your brain that kind of process timing will likely reflect those skills that you've acquired. So those are examples of your experiences kind of influencing your brain.
And I think maybe just to drive that point home is that Max was talking about expanding in the physical space. So they're not swelling, but if an area of your brain that is responsible for processing information from, say, your left hand is maybe 3 millimeters across. Maybe someone who is a professional violinist, that area is 5 millimeters across. So it's physically just takes up more space. And it could do that because the brain is plastic and dynamic, and plastic meaning changeable and fluid and malleable. And so maybe if a person who's a violinist utilizes that hand all the time and very frequently in these very fine motor movements, another area of the brain, like maybe an area devoted to processing sensory information having to do with their feet, maybe that's slightly less because the brain needed that space for the high demand that you are putting on it by playing violin all the time. The foot was just an example. It could be anything.
Right. Or like people, like if you're blind, obviously there's humans, we're very, very visual. That's kind of our main sensory modality. A huge portion of our brain, the whole back portion of our brain, is dedicated to processing all that visual information. But in individuals that aren't sighted, in blind individuals, that part of the brain isn't just going to lie dormant. It's not just going to hang out and do nothing. It's going to start getting information from the auditory system, from like the tactile system, if that individual is going to learn to read Braille. So again, plasticity gives the brain all these compensatory mechanisms that allow it to kind of adapt to injuries you may sustain over the course of your life, and also changes in your behavior. As you learn new skills, the brain will kind of reflect in its structure, will reflect the way your behavior has changed. And Emily also mentioned plasticity. It's really important from a developmental point of view. The brain is exceedingly plastic in young individuals, and actually the brain doesn't fully develop till your early 20s. So it's kind of the final part of your physical self to develop. And your experiences in your early years of life are really critical to how your brain develops. And if you take, like a classic example is an experiment where they raised rats in a really enriched environment with lots of different with lots of rats, so it was a really like social group, and they had all these toys, and then they compared those rats' brains to rats that were reared in isolation in just a box with no toys, kind of a very kind of sterile environment. And the number of neurons, the complexity of the connections that those neurons made were physically different in those two rats, showing that, to have a really mature brain that reaches its full potential, you really need there's these critical periods early in life where you need the right type of nurture. And those experiences, that kind of comforting, enriched environment growing up in, was actually like physically impacting what your brain is gonna develop into as an adult. So individuals raised in adverse conditions, they have higher probabilities of depression and anxiety, things like this, and that's because those experiences really impacted their physical self at a young age, and that's what they're carrying with them, through their life now.
It's also very variable. So there may be
And it's really very, yeah.
There may be situations where adverse conditions are faced in youth, and that doesn't have as large of an impact on the individual, just depending on genetics and maybe things we don't understand.
Yeah, there's always an exception.
There's always an exception, yeah. So, yes.
So you were just speaking about some things that are more fixed about our brain, and I think, well, that's still malleable, we can still change it. And I was listening to a podcast recently about neurogenesis, and they were giving some examples of things that you can do to actually grow more brain cells, basically, and things that were harmful and that are actually detrimental to brain cells. And I was wondering if you could give us some of those insights of some of those things that we could do, especially as some of us are getting older and,
Right.
Yeah.
doing that.
Yeah, so there's actually two really important points to discuss before we get into that. So one, throughout your entire life from the day you're born, neurons are dying, and just kind of, that just happens. It's absolutely normal. Very similar to, your skin. Actually it's not the same as your skin. So your skin, all of the cells in your skin will kind of recycle, and then over the course of maybe 30 days or something, all of your skin cells will have at one point died and been replaced by new skin cells. It's not quite the same in your brain. You're born with basically all of the neurons that you will have for your whole life, and some of them die, totally normal. Some things you do make it worse, so heavily consuming alcohol can kill brain cells. Other things can too, but it is natural, so don't worry about it. But there are also, in terms of neurogenesis, which is the creation of new neurons, in adults, there are only 2 sites in the brain where new brain cells are actually born and made from scratch. So all the brain cells that you have are not going to reproduce or make new neurons, except for in these 2 small exceptions. There's an area of the hippocampus where new neurons are made, and there's an area called the subventricular zone, which is, so inside the brain, the brain is actually not a fully solid organ. There are actually kind of holes in the inside of it. They're called ventricles, and they're kind of shaped like this. They're kind of like wings in the inside of your brain. And so in these ventricles is created cerebrospinal fluid. So this is the fluid that can be extracted from your spine during a lumbar puncture or a spinal tap, if you've heard of that or know what that is. That fluid is created and kind of supports the brain, it cushions it, helps it to be physically safe from movement damage. But on the bottom part of that region, the ventricle region, kind of down here, that's where new neurons are made also. Some of the questions in neuroscience do surround kind of why is this happening? What is the purpose of making new brain cells in these 2 regions? Does it serve a purpose underlying learning and memory, or is this something that just for some reason happens that maybe not necessarily related to a concrete function that we can point to that animals participate in? There is some research showing that the levels of neurogenesis are related to mood and depression and anxiety. One fairly easy way that you can enhance neurogenesis, which seems to be a basically good thing, is to exercise more. So healthy exercise does increase neurogenesis, at least in mice, and I think also probably in humans too. But I think it's always good to advocate for healthy exercise. So, but otherwise, in terms of how else to enhance neurogenesis, I don't really know if I have too much more advice for you.
Maybe it's not
Exercise.
important, I don't
Yeah,
know.
and it may be, yeah, we're not sure. It's something that's definitely under investigation. But it seems like a good idea, right? So.
And I think just keeping an active mind, just anecdotally,
Yes. Yes.
professors I know that are like in their late 80s and are still razor sharp, it's because I think they're just thinking all the time, they're reading all the time, they're talking and interacting, they're like not letting their life fall off in any way. They're just still so engaged, and that just keeps them really, really sharp.
Yeah. Coming to stuff like this, it's great. Yeah. You've had a question for a while. I'm sorry.
Yes, actually I remember the question.
Sure.
I wanted to ask you a couple of questions about locality and injury. My understanding is that it's possible for an area of the brain to be injured and for another area of the brain to be trained to take over the functions of that of the first area of the brain. My first question is to what extent is my mental model accurate? And if it is, are there areas of the brain that are more trainable or more trainable to certain functions than others?
Right. That's a great question. And again, that's kind of touching on this concept of plasticity, the brain's ability to kind of reorganize itself and reallocate its resources to different types of functions. So, like the example I gave of a blind individual, that's really well documented that the visual cortex of blind individuals kind of takes on other roles and takes on other functions to kind of, okay, if that person can't see, well, let's use this extra real estate to give them really good hearing and really good tactile discrimination so they can read Braille. So that can happen in individuals that have like a congenital disorder that they're born with, but it can also happen in adulthood. So if you sustain brain injury, it's all very common. This is kind of outside my area of limited, I don't even want to say my area of expertise. It's outside of what I study, so I'm kind of waving my hands. But like stroke, it's very common for individuals to have a stroke. A stroke produces a very focal lesion in the brain. It damages one very specific node. And so the consequence of that stroke is dictated by the location of the stroke, right? If it's in an area that's involved in generating speech, the person will have a problem speaking. If it's in an area that moves the right arm, the person will have a problem moving their right arm. And following strokes, I think it's common to see kind of a large impairment right away, but then as weeks, and if the person is given physical therapy and the right kind of training, you'll see those deficits kind of go away. They'll regain their functions. And it's not because that area that was damaged is being repaired. As Emily said, those cells, when they die, they're dead. Mm-hmm. So you're not seeing a damaged area recover, you're seeing compensation. You're seeing other areas coming in and saying, oh, let me help out move the arm. Instead of the information going to that now damaged part of the brain, I'll listen to what that region used to hear, and I'll take over its duties, and I'll move that arm. So again, and that brain plasticity is something that's only begun to be appreciated in the last 10 or 20 years, and it's a big focus in neuroscience now. It's really the brain is really this dynamic plastic thing, and it offers, if you can get a handle on that plasticity, there's a lot of therapeutic benefit that you can get, right? If you can control or kind of enhance the brain's ability to do all this compensation, it would allow doctors to treat mental and neurological disorders a lot better. So that's a heavily researched area, is brain plasticity. How can we improve brain plasticity? The more we can learn about it, the more we can kind of control it and use it as a tool. And parts of the brain that may not be that good at being plastic, maybe we can develop drugs that make them better at being plastic.
Yeah. We're probably not quite able to name an area that isn't good at being plastic. I don't think that it's commonly thought of that there are regions that are just not very plastic. I would say probably areas of the brain that are important for regulating your baseline function. So regulating respiration and digestion, those kinds of things I think are less plastic. Just because I think the type of tissue, so the type or the part of the brain that does that is qualitatively different than some of the other parts that are associated with things that we think of maybe more commonly associated as deficits with stroke patients, like sight or being able to hear or move certain parts of your limbs. So the brain stem is kind of exactly what it sounds like. It's the part of the brain that comes out the bottom, the brain sits on top of. That is a structure that's common to most vertebrates, and it's fairly similar in most animals. And so I think that because of the kind of long evolutionary history of that structure, there's not a lot in it that changes on a routine basis. And I think also evolutionarily it was beneficial to have a fairly stable brainstem, because if it changed too much, maybe you changed it too much and can't breathe anymore. So I think maybe that could have contributed to the kind of stability of that structure and the insensitivity to plasticity. But I'm sure that it's also very plastic. Yes?
I've come to believe that the neurons are the pathway, and the storage, if you might say, of the little bits of memory that the electrical impulses pass along that neuron, and they pick up the little tidbits and put together a picture or a conclusion or algorithm, whatever you want to call it, that give you the cognitive capability. And the astrocytes, which I would call the gray matter or the white matter, depending on what part of the brain you're looking at, seem to have, in my opinion, from what I feel like, are where the actual tidbits of memory are held. And it's kind of a temporary, constantly changing thing. Do you have an observation on what I'm speaking, the astrocyte aspect of where the memories are really in bits and pieces stored, and then the brain puts those associations, links the associations? Do you have any?
I think it's a great question, and so astrocytes are a type of glial cell that Emily was talking about in the beginning. So in the brain, about 90% of the cells in the brain are glial cells.
Yeah,
Did I
it's
just make
like
that
the
up?
very vast.
Okay, so the vast majority of
Yeah.
cells in the brain are these glial cells, of which astrocytes are one type. So there's certainly like the majority of cells in the brain. Glia means glue. They're thought to be just kind of support cells, like Emily said. They're supposed to just kind of maintain the homeostasis of the brain, feed the neurons and deal with their waste and these kind of support roles. But I'm not aware of it to a big extent, but there is a line of research, and there's a lot of researchers out there that are kind of pursuing this hypothesis that you just mentioned, where historically people have always said, astrocytes and other glial cells, they just kind of do this support work. They're not really doing anything related to behavior or cognition. There are some researchers, active researchers now, that are pursuing experiments to kind of test this hypothesis. Do these astrocytes actually contribute to memory and to cognition and to an animal's behavior? Unfortunately, I'm not that.
Yeah.
I can't think of a good example of those studies.
I don't think anybody really has.
Right, so it is certainly a wide open question. I'd say the majority of the field would say it's not the astrocytes. Where memories are embedded, where ideas arise from, it's not an individual neuron that does it, it's a circuit that does it, right? Like a computer, if I take one transistor out of a computer, it doesn't really do much. But if I put a ton of transistors together and build a computer, all this function emerges from it. So that's kind of how we look at the brain as cognition, behavior, memories. These are all emergent things. These are some magic that we still need to figure out. Something happens when all these neurons are acting in this very coherent, organized way, and they're sending these very organized signals back and forth, and they're making these connections that are getting stronger or getting weaker. New connections pop up, old connections disappear. I would say the majority of the field would say that's where cognition, that's where memories comes from. It's the connections between cells. And like we said, neurons, you're kind of born with all your neurons, and you're not making new neurons, so that's kind of like a fixed thing, right? You have a fixed number of neurons, but what you can tweak throughout your life are all the connections between the neurons. And there's really cool studies showing that you can like teach a rat or a mouse something. You could say, every time this buzzer goes off, you're gonna get some cheese. And then like within minutes of learning that, you'll see new connections in the brain, and using really fancy kind of equipment, researchers are able to watch connections form on like the second to minute time scale as you're teaching an animal something. And then if you do something to not allow those connections to be made, you'll find that the animal won't be able to learn. So I would say that all this stuff, it's really the connections and it's really the network of neurons from which behavior and memories emerge. But it's a good point about the astrocytes, and it's certainly like a big line of research that people are pursuing.
I think one thing I would add to that is that, as I mentioned, certain glial cells, like astrocytes in the brain, control the amount of ions or chemicals that are around. And one very important ion are calcium ions in the brain. They're very important for this, for the action potential. And astrocytes specifically help regulate the amount of calcium in the environment around neurons. And so there is a possibility that astrocytes regulating calcium ions could influence the information that's being processed by the cells, by the network, or the information traveling through this network by saying, oh, well, I'm going to absorb all of the calcium so no more information will be sent. And that it could totally hand-wavy here, effectively stop certain processes from happening, and vice versa. So I think that the way that neurons interact with cells like astrocytes may be extremely important for the way that information is processed and held in the brain. And so I think that's a really important question.
One last question, I'll keep it master for a while. The memories that we are forming are mostly, in my opinion, in pictures. And I think you said it yourself a little earlier, that we seem to remember better an older memory because we have some sort of a picture in our minds. And the fact that we can bring it back with some emotion attached to it, we can remember it a little bit better. It's like you put your car keys down on the counter and you explode them in your mind for 10 seconds before you walk away. You'll remember where you left them better. Or the old car accident years ago was maybe very traumatic, and that was an emotional experience. And sometimes we block parts of it out, or our heroism in solving the crisis, or helping on the crisis. But mostly it's a combination of the picture in our mind attached to some animation or motion or extremely unrealistic thing. For me, always, if I could attach a silly name to somebody's prominent feature that I look at when I'm, say, trying to meet them, if I can, and of course I never tell them what I'm seeing. You have to be circumspect, but it's a fascinating thing to me that what you said earlier about the picture we form in our mind, that's pretty much how we remember almost everything. And if we can attach motion, animation, unrealistic, cartoon-like behavior to it, we have a better chance of bringing it back more completely. And our memories tie into each other, again, this association thing. In your studies, are you looking as much at the nerves and the neurons and the astrocytes and the axon hillock and all the things, and the myelin sheath that speeds the pulse along, impulse along? Are you looking at essentially the physiology of it, or seeing, or just as also as deeply on how people can learn things?
Right. I guess most of
cognitive side, or put two and two together. So
right.
I guess maybe we could quickly, in just a couple of minutes, because I know we didn't quite get to explaining our research yet.
Yeah, actually, maybe this is a good time. Yeah, we could talk a little bit about our specific research
questions.
and then you'll see why we don't ask big questions like this.
Yeah.
Right, gentleman right there, I think he's got the same problem I've got. He's got about 50 other questions.
Well, we're happy to stay and
totally
keep chatting
happy.
afterwards. So, yeah, we'll describe really quickly kind of what we do, and you'll see in terms of, so on one end of the spectrum you have the physiology of the brain. On the other end of the spectrum you have all the weird miracle that is the mind, and thoughts and cognitions and emotions, and you're trying to draw connections and figure out like how is biology over here making behavior and cognition and memories over here. When you approach those questions, you can work with humans and put people in fMRI machines, which give you kind of images of their brain activity, and they're humans, you have language, you can ask them really complex things. You can say, remember a really emotional experience from your childhood, or try to learn this fact using this crazy mnemonic where you draw all these connections, right? And you can have a human do that. But the physiology you get is a little limited because you're just looking at these movies of brain activities from an fMRI, right? On the other end of the spectrum, you can be working with an animal like a rat, where you can put electrodes in the brain, you can turn parts of the brain on and off, you have much higher resolution, much more detailed measures of the brain, but I can't talk to a rat and say, oh, think of like the best piece of cheese you ever ate, right? So on one end, you can work with humans and really focus your power on looking at the cognition and mental side of it, or you can work with animals and really focus on the physiology of it, but then you kind of sacrifice the ability to ask a rat about cognition. Like I'll tell you about my research right now, and you'll see that what I care about in terms of the rat's behavior is which direction it's facing in the world. So that's pretty easy to determine from a rat, right? So I guess with that, let me tell you about my work, and hopefully these limitations and this continuum becomes a little more illustrated. So we have like a sense of vision and a sense of audition, right? One sense that we always take, that most people take for granted, is like a sense of our location and a sense of direction in the world. This is something that you refer to as spatial cognition. So we learn the physical environments that we live and work in, right? We kind of form what you'd call a mental map. So you can think of your house and you have this kind of, you can bring up this map of what your house looks like, what your work environment looks like. And anytime, like at the snap of the fingers, when you're in your house, you know where you are in the house, you kind of know which direction you're facing. You know if I need to go to the kitchen, you know what direction to turn and what direction to walk to get there. And that's something that's kind of subconscious, right? You never really actively form mental maps. Maybe if you're in a new building, you may try harder to like build a little map of it so you know how to get out, but you kind of just build this spatial knowledge. And this is what allows you to navigate in the world. So I'm trying to figure out the circuits in the brain that are responsible for kind of processing and dealing with all this spatial information, and the circuits that are ultimately going to give us a sense of direction. So to do that, I work with rats, and this allows me to put electrodes into their brains, which allows me to measure these spikes, action potentials, from single neurons, which you really can't do in a human. So now I have a rat, and I have a little electrode in it, and I can measure spiking of individual cells, and I can put these electrodes in very specific parts of the brain. So say I think, oh, this part of the brain, region X, is really important for this spatial stuff. Let me put an electrode there and see what cells are doing. Let me measure these spikes. So when you do this, you find these things called head direction cells. And what you find is if you record, the technology is such that you can record spikes from a freely moving rat. You can have a rat like in a maze doing some task, running around, and you're looking at these spikes from individual cells. And there's these cells called head direction cells, which will fire these spikes. These cells will become active only when the animal is facing one particular direction in the room. So say the cell will be totally quiet, totally inactive, but now when the rat faces this direction, that cell goes crazy. I can record tens or hundreds of these neurons at once and find that some cells like this direction, another cell likes this direction, another cell likes that direction, such that no matter what direction the animal is facing in the environment, there's a unique subset of head direction cells that are active. And I'm gonna have to just wave my hands, but you can see how like when you see like a signal that robust and that compelling in the brain, maybe the fact that I know what direction I'm facing is somehow arising from the activity of cells that behave in this kind of directional way, right? So maybe I know what direction I'm facing because I have these head direction cells. So you can do all sorts of really cool tricks for these cells. They care about visual cues and also about like internal cues. Let me just draw a quick picture. Does this make sense? Do people get these cells? It's hard to visualize without like a picture of what these experiments actually look like. Generally we have like a cylindrical environment, so it's just like if you imagine kind of like a big drum, and we have our rat in here, and he has little LEDs attached to his head so we can track his position, and the rat's just running around. And while the rat's running around, we're recording what individual cells are doing. And we may find that there's a cell that fires these spikes only when the rat is facing this one direction in that cylinder. And what we do in this cylinder is we put a big like piece of white paper on one side to kind of be a landmark. So it like visually distinguishes one direction from another direction, right? Just like if you use like a clock tower in town as a landmark. So you know when you're facing the clock tower that, oh, downtown is that direction, right? We give that rat that landmark, and what you can do is say this cell fires, it would be easier if I do it this way. Say this cell happens to fire in that direction. Again, there'll be cells that are tuned to every possible direction. This is just one cell. We can record that cell, it's firing only when the rat faces this direction. You can take that animal out, kind of sneakily go in there and now rotate that landmark, and now put the landmark in a new position. I rotated the landmark this direction. So now the cell, it won't fire for this direction, now it will fire when the rat faces that direction. So it's kind of preserving its relationship relative to this cue, right? So this is showing that these cells that really want to know what direction the rat is facing, they're paying attention to these landmarks in the environment, which makes sense because that's kind of how we'll intuitively navigate.
Is that visual?
Yeah, so that's visual.
That's what they see.
It's what they're seeing.
Yeah, okay.
But the
Do you
real
ever blindfold them?
Great question. Okay, yeah, you should come work in the lab. So that's the obvious next question, right? Well, do they need this visual information? So you can take out that landmark, you can blindfold them, turn off the room lights, just eliminate all visual input, and the cells are still fine. They'll still maintain that directional firing. They'll be a little worse, maybe they'll fire for this direction or this direction or this direction, but so their tuning kind of degrades a little bit, they get a little noisier, but they're still very directional. And that's because like I can close my eyes, right? Like I have no visual input at all, and like you're there-ish, right? So how can I do that? That's because we have, in addition to visual information, these cells are also getting all this input from the vestibular system. And so in our inner ear, we have these specialized structures that detect our movements. So when I'm spinning, I know, okay, I'm spinning, clockwise at approximately like this speed. And so a really smart cell can process all that information from the vestibular system and keep track. And that cell can, again, no visual information. It can be like, okay, I'm firing, this is the direction I like. Oh, okay, my vestibular system says we moved, so now I'm not gonna fire. Oh, okay, so now I've moved about a full 360 degrees, I should probably fire again, and it'll keep firing. So these cells are what you would call multimodal, meaning they don't get input from just one sensory modality, but they're getting information from the visual system about landmarks, they're getting information about the animal's movements from the vestibular system, and there's even like other inputs coming into these cells. And they're doing some miraculous computation, they're like this little computer that's like, okay, based on the fact that the landmark's over there and I had just turned about 90 degrees, I should probably be facing around this direction.
Do you ever put them in a centrifuge and then test them? Really spin the hell out of them?
Yeah, so there's different ways to disorient them. We've done like manipulations of the vestibular system, and that seems to like the cells really can't do much without that vestibular input. That seems to be most critical.
Once it settles down, they recover.
Yeah, so you can, if you like permanently, if you temporarily kind of turn off the vestibular system, there's a bunch of ways to do that. You can simply just like put some drugs in the inner ear, and you can kind of turn off those receptors in the vestibular system. The cells get wonky, they don't really know how to behave anymore, they just start firing spikes for all directions. But then as that inactivation wears off and the vestibular system comes back online, the cells go back to doing what they do.
Do you hypothesize that most of us don't know where the hell we're going most of the time?
Right, everybody always says, well, I don't have these cells because I have a really bad sense of direction.
But there are people throughout history who have been able to navigate across the strangest, surfaces, ice caps, people who were able to walk across deserts, and they know where they're at.
Yeah, so this is, kind of a cool example to touch on that is, so in addition to these head direction cells, which are kind of like a compass, we think of this as like the brain's compass, the brain also has a map. There's these cells called place cells. They don't care about the direction that the animal's facing, they care about where the animal's located. So one place cell will activate and fire spikes only when the animal is in this part of the room. And then the animal moves and that cell goes quiet. But then another cell turns on for this location. Then the animal moves again, okay, those cells are off, but there's a cell tuned to this direction. And again, we think that it's the activity of these specific cells that ultimately turns into your perception of, oh, I'm kind of in this part of the room and I'm kind of facing this direction. I think that because those specific place cells are currently active and those specific head direction cells are currently active. So I introduced place cells because they're found in this part of the brain called the hippocampus. And there's this really cool study done by some researchers in London where they looked at taxi cab drivers. And you can imagine a taxi cab driver, particularly a London cabbie, they have to take this exam where they need to commit the entire city to memory and be able to like verbally rehearse routes between any two locations. So they build this phenomenal map in their brain and they spend their career accessing this map to do their job. So these researchers said, okay, well, let's look at the hippocampus. That's where these place cells, these map cells are. Let's look at the hippocampus in normal control individuals and compare it to what the cabbie's hippocampus looks like. And they found that the cabbie's hippocampus was significantly larger than someone who doesn't navigate for a living. And then they even looked at retired cabbies, and their hippocampi had kind of contracted back down to a normal level as they kind of stopped navigating every day. So there is, and that gets back to this nature versus nurture, like that study would suggest that the brain differences result from the behavioral differences, not vice versa. But there's the parts of the brain that have these cells. There's some very rare conditions in which those parts of the brain are impaired. There's just damage to those regions congenitally. And these people are basically born without a sense of direction. So certainly there's evidence that these systems can be disrupted. And when they are, your kind of sense of direction is super impaired or non-existent.
Are there people without any proprioception?
I don't know.
Yeah.
There may be disorders in terms of like feeling your like limbs in space.
Yeah. Like people with developmental disabilities that are congenital.
That can selectively
So-called
affect.
autism is such a global thing. It's such a
Right.
basket. But some people,
Oh,
and your research has profound possibilities for that,
Right.
for helping people.
Right. And we know, and so proprioception, like feeling, that's like our sense of how our body is positioned, where our limbs are my arms overhead, or are they at my sides? We have receptors in all our joints that kind of detect the position of our body. And we know that those systems also influence these head direction cells, because head direction cells act differently. If the animal's executing its own movements and like freely moving, they work just fine. But if you move an animal around passively, meaning you just like put an animal in a little cart and now you move it, the visual experience is the same, but it doesn't have that proprioception. It's not generating its own movements. Then the cells you see, they kind of get a little wonky. They kind of don't operate fully, showing that that's yet another type of input that this circuit gets. So it's getting visual information, it's getting vestibular information, and all this proprioceptive information.
Yeah, really important.
So do we have maybe just, are we almost done or done or over?
A little
We're done?
time for yours.
Okay, great. Yeah, I'll do it really quick. So I actually kind of explained how I do my work already. So when I was talking about the neuron, the technique that I use to ask questions about the brain involves recording the electrical activity of single neurons, very similar to Max, but I record neurons in slices of brains. And this allows me to record a neuron that I can see. So that's different from the way Max does it. The neurons that Max records are inside of the skull of an animal that's awake and doing something. In a situation when the animal was implanted with an electrode. But in my situation, the brains have been taken out and I look at the cells underneath the microscope. And that's important because cells look different in the different regions that they are a part of. And there are different subpopulations of cells within those regions that are involved with these things called circuits. And so Max talked about circuits a little bit, but kind of broadly as just a definition of what we think of as circuits are different parts of the brain and different cells in parts of the brain that work together to accomplish a goal. And so some of the circuits that I look at exist in the prefrontal cortex, which is the area of the brain that was damaged in poor Phineas Gage. And the cells there send their axons to different parts of the brain. And based on where they send their axon, that's kind of how we subclassify these cells. So there are some cells that talk to your brainstem, and we call those brainstem projection cells. And then there's cells that talks to other areas of the brain that are kind of on the surface. The top part of the brain is called the cortex. That's the kind of bumpy part that you kind of think of when you first think of what the brain looks like. So cells that send their axons to different parts of cortex are different from cells that send their axons to the brainstem. And I look at those two types of cells, which I can see under the microscope, and I measure their action potentials, which is that electrical impulse. I measure their general physiology, so kind of what they're like, kind of what they do on a daily basis. Are they really responsive to neurotransmitters? Are they not responsive at all? Do they have different proteins in their membrane that manipulate and change the types of the shape of the action potential, how effective an action potential is, that kind of stuff. So one of the kind of important questions that I've been investigating that Amanda talked about early on is how do different therapeutic drugs that people take, like antidepressants, affect those systems that I just described. So we were talking about how neurons express many types of receptors, as we had a question about. And there are certain drugs that can change the amount of neurotransmitters that affect those receptors in the brain. So many mood disorders, like depression and anxiety, bipolar disorder, obsessive compulsive disorder, these types of things are thought to relate to the serotonergic system, which is essentially just the system that releases serotonin, the neurotransmitter, throughout the brain. And it's not clear if there's not enough serotonin or too much serotonin. That's always the case. But the reason that we think that serotonin is involved in these disorders is because in some patients, taking a therapeutic drug like an antidepressant, especially one called a selective serotonin reuptake inhibitor, or an SSRI, can increase the amount of serotonin in the brain, and some of these patients feel better. So it kind of led us to the hypothesis that, oh, well, maybe in some of these patients with these diseases, they're suffering from not enough serotonin. So when we give them this drug, it makes them feel better. Great. In only about a third of patients with mood disorders like depression and anxiety actually respond to drugs like SSRIs. But in those cases, we are still not sure why more serotonin means that people feel better. And the reason that is because when you take an SSRI, immediately in your system, there's more serotonin. These drugs work really fast. So probably within the hour of taking that medication, you would be able to measure more serotonin in your brain. However, patients that are responsive to these drugs don't feel the beneficial effects for 6 weeks, 3 to 6 weeks on average. And that's really baffling to us because we know that the drugs work fast, but they don't feel better for a while. So is there really a connection between serotonin being high in the brain and feeling better or not feeling better? So one of the questions that I've tried to understand is, are there changes that happen over time in neurons that could potentially lead to these beneficial effects of these drugs, that the types of things that may change that make us feel better? And one of the results that I found is that in these two groups of cells that I originally referenced, the cells that talk to other areas of cortex and the cells that talk to the brainstem, the responses to serotonin in these cells in rats are the same. They're both enhanced. Their action potential number is enhanced when serotonin is around in rats. However, after giving these animals Prozac for 3 weeks, what I found is that one group of the cells that send their axon to the brainstem, are actually now turned off by serotonin, while the other group stays the same. And so their responsivity...
Stay the same, meaning
they're still
action
excited.
potential is greater.
Yes, more action potentials, exactly. So these responses to serotonin are mediated by different receptors. Like I said, you could have the serotonin hooking up with one receptor on the membrane that says yes, go more, or another type of serotonin receptor that says no, go less. That seems to have changed in the brain of rats at a specific dose. This may not happen in humans, so please be wary of that. But what we think, the kind of take home from this is that a lot of drugs that alter neurotransmission, so the amount of serotonin in the brain, amount of other types of neurotransmitters in the brain, may be having effects on neurons, even on neurons that are not producing serotonin, and that these effects may be develop over time and may last a really long time, and that those may be the actual thing that helps us to feel better. So that it's not just, oh, well, a simple, you need more serotonin, there you go, you feel better, but that there may be this delicate balance of different types of proteins or receptors that respond to neurotransmitters in a specific way that is changed. And that by giving these drugs that affect the serotonergic system in this case, we can change that balance. And that maybe that is helpful to some patients. It's very, very preliminary. We have no idea if this would be something that we could test in humans, but it starts to kind of help us think about these diseases in a slightly different way. And it also kind of teaches us more about how systems in the brain are, again, plastic. So not just the synapses, not just the connections between them, but maybe at the level of what proteins are most important to these cells that actually result in different kinds of activity. Maybe those kinds of things can change too, and that can change with age or with experience or with exposure to different drugs like therapeutic drugs or other types of drugs. And so that's something that I study in rats. I've looked at some behavioral stuff with them too, so putting them in mazes and boxes and seeing if they can learn better or worse with the Prozac in their system or otherwise. But I ask a lot of questions about how cells work at the very small level. And I am I analyze microcircuits, and whereas Max might call himself a systems neuroscience that understands kind of a neural system rather than an individual one circuit. Though you can study circuits and singular nodes of circuits that are involved with the systems that you're studying. So, yes.
So to that end, I have a question about how the thought process with what you just said, what you're studying.
Yeah.
How the mind and the thought process, from what I'm understanding, in a relationship said with past trauma, how you can train, what I'm understanding is the neurons or the chemistry.
Yeah.
To perceive and react or to shut off the flight instinct,
Right.
like PTSD.
Yeah.
Right? And you can, so it
Right.
seems to me that's like a chemistry, but I'm curious about what your thought is about how does that, the thought process of everything going on, it seems to me it somehow can tell certain cells to not fire, given a certain situation, a memory.
Right.
You know, that you alluded to in your previous, you know.
Yeah. So PTSD is post-traumatic stress disorder. It is characterized by an exaggerated fear or stress response to what is now a fairly benign stimulus. So someone who has had an experience in a situation like war, where they're exposed to a lot of noise, like gunshot noises and explosive noises resulting in extremely bad situations, traumatic experiences. Now, in a situation like this, maybe if someone dropped a book unexpectedly, that very loud bang noise may evoke a very a heightened stress response in that person. And there is both a thought, the kind of a cognitive and thought level to that response, and also the physiological response. And we kind of alluded to this earlier with your question, how there is a relationship between our certain thought processes and the physiological feeling that we have in our body. And so I think some of the issues that are going on with PTSD is that there are now, as a result of certain levels of trauma, or certain types of trauma, there are newly formed, very strong connections between different cells in the brain or entirely, full regions of the brain that normally were not, or beforehand were not so strong. and that it's very hard to break those down. Actually, this is referring to, I think, maybe you, something gentleman in the back was referring to earlier, how emotions and experiences can influence our memory. So something that's very, very emotional usually, in some cases, can form extremely strong memories compared to something that's a little bit more, less emotionally charged. And so in those situations where you have that heightened connection, that's building of a much stronger connection, you both have thought processes, for example, flashbacks and recurrent memories that are very, feel very real and very, very poignant to the person. And that's kind of like a cognitive process that goes on, which then does lead to that, that physiological change. And I think a lot of the work that's done in trying to understand how we can help patients with PTSD is going into kind of understanding how we can break that connection and reduce that. And so sometimes therapeutic drugs are prescribed to patients with this disorder. And my understanding is that a lot of these drugs are not super effective in helping these kinds of things. So we think that outside of, it may be a little bit more effective to work on the thought side, breaking that connection. So working with patients to kind of use something what's called extinction therapy, where a patient is exposed to something that's fearful to them at kind of maybe at a more a lower level, and then continually re-experiencing that kind of benign version of something that produces this heightened fear, to then slowly make your body realize, oh yeah, that's not so fearful, that's not so scary. And in terms of how that relates to neurotransmitters making more action potentials or less action potentials, I would say, at least myself, I'm not sure how much we know about that. There are actually some cases where fewer action potentials is good, and that actually sounds counterintuitive, but can promote certain behaviors because of the way that circuit is set up. So there may be one part of the circuit that needs to be quiet in order for another part of the circuit to be effective. And so in terms of kind of saying, okay, well, if you have PTSD, maybe you have too many action potentials in that fear circuit, let's just turn them down. I don't know if that would work, because that's actually what some anti-anxiety drugs do. So certain drugs that are kind of they're called downers, kind of, what is the word? Like kind of in conversation, they may be called downers or benzodiazepines. Those are types of drugs that will just reduce the number of action potentials throughout most of your brain. It just kind of turns everything down, kind of calms you down, depresses the nervous system. And for some people with PTSD, that doesn't actually change anything. So we're not really sure if more or less action potentials is what's important in those specific systems. Did I answer your question? Do you have another question?
No, well, in my experience, personally, I've been able to train myself from a traumatic situation. And I've been able, and it's just a mystery to me. I mean, it works.
Yeah.
But I can't explain how it works. This is why I'm intrigued with what you're studying.
Right. Yeah.
You know, how your emotion, how you can train your brain to recognize those situations that maybe you have a strong connection.
Right.
That's going to, quote, flip the trigger.
Right.
And how you can recognize, how you can teach your brain. So I'm wondering if my question is how you can teach your brain to recognize that and say, no, that's not what happened, this is what's happening. And then your whole system changes when you recognize that. And I'm just wondering if it's that neuron, kind of the chemicals, that somehow your emotions, what we call emotion, is just reorganizing the chemistry, teaching your brain how to react to the situation. I don't know, that's kind of vague, but.
No, I think I understand a little better. So the parts of your brain that control emotion are kind of evolutionarily older, and so what that means is that many animals share those parts of your brain, and then they're kind of, you know, a lot of animals have them, and they're controlled by more complex and advanced parts of your brain. So to a certain extent. So the cortex is, like I said, that top part of the brain, and there are areas of cortex that perform what we call top-down control, and it kind of physically means that cells out on the top control the cells kind of on the bottom, but it also more abstractly means that cells that are involved with cognition and emotional regulation, which is actually specifically the part of the brain prefrontal cortex. With Phineas Gage, he was not so good at controlling his emotions. I'm not referring to Max, he told the story.
I can control my emotions like a big boy.
So that area of the brain prefrontal cortex is involved with this top-down or higher level processing control of the parts of the brain that mediate our emotions. So an area of the brain like the amygdala, which is located close to the hippocampus, and you have one on each side, that's a part of the brain that's very important in applying kind of emotional valence, so saying whether or not something is very important, we should remember this, it's really exciting, or it's very scary, very fear-inducing. The amygdala is involved with that, and it's classically thought to be mostly involved with fear, but we are finding more out about how it helps us understand that some things are very important, happy emotions too. But that area of the brain is thought to be one of the nodes that's having trouble in PTSD. And you may be able to train your cognitive processes, maybe in prefrontal cortex, to talk and send signals down to your amygdala to say, okay, right, you want to have that response. You are getting these sensory input that makes you have this flight response, this high anxiety or high stress state, but it's okay. We can control that. We can calm ourselves down, and it's kind of this different levels of control can be more finely tuned or strengthened to kind of physically control those responses. I mean, anyone who's experienced something that was actually ended up turning out to be benign, so maybe like touching a stove that you thought was hot and pulling your hand away and then feeling, oh, that was scary, I could have burned myself, but I'm actually fine. You can kind of breathe deeply and think like, oh right, that didn't happen, it's okay, and you can calm yourself down. And that's part of that, those kind of more complex processes coming in and maybe helping to control. And parts of the therapy that's employed by counselors and therapists does kind of try to play into this situation. So we were talking about the extinction therapy, where someone is exposed, or I'm sorry, I think I called it exposure therapy before, where someone is exposed to kind of that stimulus multiple times to kind of teach their brain, right, it's not as stress-inducing, it's not so scary, it's okay, you don't need to have this heightened response to this. That may be training that top-down control, and specifically training cells in your prefrontal cortex to have tighter control over that. There may be physiological changes that are detectable in that part of your brain before and after therapy if it works. So
yeah, through the process.
yeah, absolutely.
So I think we're gonna wrap up.
Yeah.
Certainly.
Yeah, I think we've
Covered a
long
lot of
time.
ground. So again, thanks so much to Emily
Appreciate it.
and
Thank you
Matt.
guys so much.